Hello friends. Today we are going to discuss about Calvin cycle in detail. Calvin cycle in detail. Green parts of the plants, green parts of the plants consisting of chloroplast, and in this chloroplast we can see a transparent solution, and this transparent solution is known as stroma. In the stroma we can observe a molecule called rubisco. A molecule called RUBP. RUBP means ribulose 1,5-bisphosphate. It is also called as RUBP. So this is a molecule present in stroma, and this molecule having the affinity to absorb carbon dioxide. Affinity to absorb the carbon dioxide, and this RUBP is 5-carbon compound. 5-carbon compound, and carbon dioxide is 1-carbon compound because it's consisting of 1 carbon atom in it. So when these two molecules combine together, 5-carbon compound plus 1-carbon compound, it produces 6-carbon compound. 6-carbon compound, and the 6-carbon compound is an unstable compound. Because of its instability, after its formation, immediately it splits into two molecules. Immediately it breaks into two molecules, one with 3-carbon compound and another one with 3-carbon compound. So the 6-carbon compound is splitting into 3-carbon compound and 3-carbon compound. So one molecule is splitting into two molecules now. One molecule is splitting into two molecules. And you have to understand one more concept here for preparation of one glucose. Why we are tracking this Calvin cycle? Because how glucose is formed. Okay, how glucose is synthesized. So on that basis, we are seeing this Calvin cycle. So to prepare one glucose, to synthesize one glucose molecule, 6 carbons are required. If you see the formula, chemical formula of this glucose, it is C6H12O6. C6H12O6. So here we can see that 6 carbons are required to synthesize one glucose molecules. But here we take only one carbon dioxide. Here we take only one carbon dioxide. So in order to fix one glucose, how many carbons are required? 6 carbons are required. So we have to take 6 carbon dioxide. How we can take 6 carbon dioxide? Because one RUBP means ribulose 1,5-bisphosphate accepting 1 carbon. So 6 RUBP accepts 6 carbon dioxide. 6 RUBP accepts 6 carbon dioxide, and the byproduct which is produced here is 6 molecules of 6-carbon compound. Right, 6-carbon compound, and this 6-carbon compound immediately it's splitting into two molecules. Right, here I am writing here 6 molecules are splitting into 12 molecules, right, each with 6 molecules, splitting into 12, each with 6 molecules, and these are 3-carbon compounds. The 3-carbon compound which is produced here is 3-phosphoglyceric acid. Don't confuse here 3 means the position of the phosphate, So 3-phosphoglyceric acid, that's what we seen here. See, 6 molecules of RUBP when combined with the 6 carbon dioxide, it gives 12 molecules of 3-phosphoglyceric acid. And it is a 3-carbon compound, whereas RUBP is 5-carbon compound and carbon dioxide is 1-carbon compound. This is catalyzed by the enzyme rubisco. And you have to know one more thing, that rubisco means ribulose 1,5-bisphosphate carboxylase and oxygenase. Ribulose 1,5-bisphosphate carboxylase oxygenase. Here I am using two words, that is carboxylase and oxygenase. This rubisco enzyme having the affinity, having the ability to absorb both carbon dioxide and also oxygen. I'm repeating again, rubisco having the ability to absorb both carbon dioxide and oxygen. So in atmosphere, if carbon dioxide is more, then rubisco absorbs carbon dioxide. In atmosphere, if oxygen is more, then it will absorb oxygen. So you will get one dot. If both are in equal proportion, then what will happen? This rubisco absorbs carbon dioxide because it is more affinity towards carbon dioxide when compared to oxygen. It will give first preference to carbon dioxide. If rubisco absorbs carbon dioxide, it undergoes Calvin cycle. It undergoes Calvin cycle. If Rubisco absorbs oxygen, then it will undergo photorespiration. Photorespiration, which we will study about this photorespiration in further class. In further class. So we know that the carbon is fixed into the cycle. The carbon is fixed into the cycle now, and this process is known as carboxylation process. And this process is known as carboxylation process, which means I'm writing here carboxylation process, which means fixing of carbon dioxide. Fixing of carbon dioxide. And the next one is the 12 molecules of phosphoglyceric acid, the 12 molecules of 3-phosphoglyceric acid undergoing phosphorylation. I'm repeating again, these 12 molecules of phosphoglyceric acid undergoing phosphorylation. Phosphorylation means adding of phosphate group, adding of phosphate group. Already we having phosphate at third carbon in this 3-phosphoglyceric acid molecule. Again we are adding phosphate group. That's why now we are seeing that at first carbon and also at third carbon phosphate is added. Phosphate is added. How many molecules are there? 12 molecules are there. So we are taking here 12 molecules of 1,3-diphosphoglyceric acid. Why we are using di? Because 2 phosphate group are present in this. 2 phosphate group are present in this. How this extra phosphate is added? Through ATP. Through ATP. So how many molecules are added? 12 ATPs are added. So here we can see for 12 molecules, 12 ATP are added. After this, it converting into 1,3-diphosphoglyceric acid, diphosphoglyceric acid, and this is catalyzed by the enzyme kinase. This is catalyzed by the enzyme kinase, and this process is known as phosphorylation. Phosphorylation, and this after the phosphorylation, this 1,3-diphosphoglyceric acid is hydrolyzing. Hydrolyzing. So what happened? NADPH2 is added here. 12 molecules of NADPH is added here because 12 molecules are there. So 12 NADPH, here it is 12 NADPH. 12 NADPH. Sorry. Yeah, here 12 molecules of NADPH2 is added to the cycle and releases NADP and releases NADP. Releases NADP and it forms phosphoglyceraldehyde. See here acid is converting into aldehyde after dehydrogenase, after dehydrogenation process. After dehydrogenation process. So what happened? Acid is converting into aldehyde now. The product which is produced here is phosphoglyceraldehyde. How many molecules? 12 molecules. Why it is? It is also a 12 molecular compound and there is no change in the carbon, right? That's why this is also a 3 carbon compound and this is also a 3 carbon compound. The assimilatory powers which are generated in the light reaction before this Calvin cycle, there is a light reaction. In that light reaction, the end products are ATP and NADPH. From that ATP and NADPH, we are plant utilizing that assimilatory powers like ATP and NADPH in this cycle. And the NADP which is produced here, again it entering into the light reaction and it helps in absorption of hydrogen, and again the process is going on. Now here we got 12 molecules of phosphoglyceraldehyde. This is also called as glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. This is also a 3 carbon compound. To know the fate of this 3 carbon compound, okay, before knowing of this fate of this 3 carbon compound, phosphoglyceraldehyde, before we studied that phosphorylation process and also that is dehydration process, that is dehydrogenase process. These two processes together called reduction process. The first process is carboxylation process and the second process is reduction process. In reduction process, we can see both phosphorylation and dehydrogenase process. Right now, we will see the fate of this phosphoglyceraldehyde. So now we having 12 molecules of phosphoglyceraldehyde here, and this for our convenient to understand how this process is taking place, we splitting these 12 molecules into four groups. Yeah. See here, one with two molecules, one with five molecules, one with three molecules, and one with two molecules. 2 plus 5 it is 7, and 7 plus 3 it is 10, and 10 plus 2 it is 12. So 12 molecules we split into four groups, out of which 5 molecules of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is a 3-carbon compound, and it undergoes isomerization. And forms into dihydroxyacetone phosphate, and it converts to dihydroxyacetone phosphate. Simply we can call it as DHAP, dihydroxyacetone phosphate. This dihydroxyacetone phosphate, see here 5 molecules are there, and these 5 molecules are undergoing isomerization. So it converting into DHAP. What is meant by isomerization? Isomerization is nothing but it is a process in which glyceraldehyde 3-phosphate is converting into DHAP, and again DHAP having the ability to convert back into glyceraldehyde 3-phosphate. It can change its reaction. It can be vice versa. Process. That process we will call it as isomerization. So glyceraldehyde 3-phosphate can be converted into DHAP, and again this DHAP can be having the ability to convert back into glyceraldehyde 3-phosphate. That is what isomerization process, and the enzyme which is catalyzed this process is isomerase enzyme. So 5 molecules here, DHAP. Again we are splitting these 5 molecules of DHAP into two groups, because for our convenient purpose. So again we are dividing this into 2 molecules of DHAP and 3 molecules of DHAP. This DHAP is also a 3-carbon compound. Why? Because glyceraldehyde 3-phosphate only is converting into DHAP, dihydroxyacetone phosphate, right? So here 3-carbon, glyceraldehyde 3-phosphate is a 3-carbon compound, whereas DHAP is also a 3-carbon compound. So here this is a 3-carbon compound, and this is also a 3-carbon compound, out of which 3 molecules of DHAP undergoing condensation. I'm using a word called condensation. I'm repeating again, I'm using a word called condensation. This 3 dihydroxyacetone phosphate is condensed with 3 molecules of glyceraldehyde 3-phosphate. We use 5 glyceraldehyde 3-phosphate here. Now we are using 3 molecules of glyceraldehyde 3-phosphate here. Glyceraldehyde 3-phosphate is a 3-carbon compound. Okay, and DHAP is also a 3-carbon compound. And now I am using a condensation, the word called condensation. See, condensation means 2 molecules are condensed into 1 molecule. So here 3 molecules are there, and here 3 molecules are there, and finally they are condensed into a 3 molecular compound. But carbons are not combining into a single compound. Carbons are present as such. So 3 carbon plus 3 carbon, it became a 6-carbon compound. Molecules are 3, but carbons are doubling here. You have to understand this. And this is known as condensation process. And the enzyme which helpful or which catalyzes this process is aldolase enzyme. You have to remember this aldolase. It is very important. Why? I will tell you later. Aldolase enzyme. Okay. What happened? 3 molecules of fructose 1,6 bisphosphate is produced here. Fructose 1,6 bisphosphate is produced here after condensation process, and this is a 6-carbon compound. We know that here 3 carbons are there, and here also 3 carbons are there. This is what condensation process is taking place here. And this fructose 1,6 bisphosphate, again we are splitting into 2 molecules, that is 2 molecules of fructose 6-phosphate and 1 molecule of this fructose 6-phosphate undergo phosphorylation. Phosphatase. Phosphatase means removal of phosphate. See here, at first carbon we having phosphate, and 6th carbon also we having phosphate, right? So after phosphatase, phosphate is releasing. So what happened? Fructose 1,6 bisphosphate is turning into fructose 6-phosphate. And it is also a 3 molecular compound, because these 3 compounds are undergoing phosphatase through an enzyme called phosphatase enzyme, and it transferring into fructose 6-phosphate, which is 6-carbon compound. Again, this 6-carbon compound. There is no change in carbon, right? And for our convenient purpose, again these 3 molecules of fructose 6 phosphate, we are splitting them into 2 molecules and 1 molecule. Out of these 3 molecules, 1 molecule is helpful in the formation of glucose. 1 molecule is helpful in the formation of glucose. So glucose is also 6 molecular compound, right? This is also a 6 molecular compound. And remaining 2 molecules we having now. Remaining 2 molecules of fructose 6 phosphate, and this is a 6 carbon compound. And here now we are taking this 2 molecule of 3 phosphoglyceric acid. These 2 molecules of 3 phosphoglyceric acid. Okay, now we are bringing this here. We are bringing this to here. Okay, now here we having 2 molecules of fructose 6 phosphate and 2 molecules of glyceraldehyde 3 phosphate. Here one interesting topic is, out of this 6 carbon, 1 carbon is giving to this 3 carbon compound. Out of these 6, 1 carbon is given to this 3 carbon compound. So here it will become 5 carbon, and here I will change color, you will understand easily. Yeah, here it will become 5 carbon, and here it will become 4 carbon. So now what happened? The 5 carbon compound which is produced here is known as xylulose 5 phosphate. After losing of this carbon, it turning into xylulose 5 phosphate, and after gaining of 1 carbon here, it turns into erythrose 4 phosphate. And here 2 molecules are there, and here 2 molecules are there. In this process, 2 byproducts are producing. In this product, 2 byproducts are in this process. 2 byproducts are producing here, and these 2 byproducts are produced because of the catalyzation of an enzyme called transketolase. I'm repeating again, transketolase. Why? It is transferring keto group. 1 carbon it is transferring, right? That's why it is called as transketolase. Now you have to understand that 1 aldolase processes happen, 1 phosphatase processes happen, and another one is transketolase processes happen. So what happened? Again this process repeats, means 1 aldolase have to be takes place, and 1 phosphatase have to be takes place, and transketolase have to be takes place. See, now here we having 2 intermediate compounds. Sorry, not intermediate compounds. Yeah, 2 byproducts are produced. That is xylulose 5 phosphate, which is a 5 carbon compound, and erythrose 4 phosphate, which is 4 carbon compound. This erythrose 4 phosphate, which is 4 carbon compound, undergo again condensation with 2 molecules of DHAP. Which is produced here. Okay, these 2 molecules of DHAP and these 2 molecules of DHAP. I told you that condensation means these 2 molecules will combine into a single molecule, but carbons won't be combined. Okay, carbons will be present as such, right? So here 4 carbons are present, and here 3 carbons are present. Total 7 carbons will be produced. So the formed compound is 7 carbon compound, which is pseudoheptulose 1,7 phosphate. So how many molecules here? 2 molecules are there. The 2 join together, the 2 condense together form 2 molecules of pseudoheptulose 1,7 phosphate. It is a 7 carbon compound. So after aldolase, what happened? Phosphatase have to be happen. So this pseudoheptulase 1,7 phosphate undergoing phosphatase. Phosphatase means what? Liberation of phosphate. So after liberation of phosphate, it turns into pseudoheptulose 6 phosphate, because it losing 1 phosphate. Phosphate group. So here 2 molecules are there. So here also 2 molecules will be present. After that, what happened? Transketolase have to be happen. So this 7 molecule of pseudoheptulose 6 phosphatase combining with here 2 glyceraldehyde 3 phosphate is present at starting, right? So this 2 molecules of pseudoheptulose is a 7 carbon compound, and 2 carbons are given to this glyceraldehyde 3 phosphate. Then what happened? This 3 carbon compound is turning into a 5 carbon compound. Okay, the 5 carbon compound which is produced here is ribulose 5 phosphate, ribulose biphosphate. And after liberation and after giving off two carbons, how many carbons will be present here? 7 minus 2, that is 5 carbons will be present. The 5 carbon which is produced here is xylulose 5 phosphate, xylulose 5 phosphate. Again it producing xylulose 5 phosphate. So in this process, two molecules of xylulose 5 phosphate is produced, and in this process also 2 molecules of xylulose phosphate is produced. So total 4 molecules of xylulose 5 phosphate is present here. Total 4 molecules of xylulose 5-phosphate is present here. And another molecule which is produced here is ribulose 5-phosphate. It is also a 5 carbon compound. Don't confuse Amma. In transketolase enzyme, during the transketolase enzyme, if this enzyme catalyzes means two byproducts will be produced. Two byproducts will be produced. Here the two byproducts which is produced is xylulose 5-phosphate, yeah, xylulose 5-phosphate and erythrose 4-phosphate. But here two byproducts which is produced is ribulose 5-phosphate and xylulose 5-phosphate. So here 2 molecules are there. Already 2 molecules are produced here. So total 4 molecules are present here. Total 4 molecules are present here. 4 molecules of xylulose 5-phosphate and 2 molecules of ribulose 5-phosphate is present here. And this ribulose 5-phosphate undergo isomerization. You know what is meant by isomerization, right? So this undergo isomerization and forms ribulose monophosphate. Ribulose monophosphate. So how many molecules are there here? 2 molecules, and these 2 molecules are turned into ribulose monophosphate. And here we having 4 molecules of xylulose 5-phosphate, and this xylulose 5-phosphate also undergo isomerization and forms ribulose monophosphate. Through this process, 2 ribulose monophosphate is produced, and through this process 4 molecules of ribulose monophosphate is produced. And here this process is known as and this process is catalyzed by the enzyme epimerase. Epimerase in the sense, see, you have to understand, in isomerase means it can be interchangeable. It can be interchangeable. Epimerase in the sense, based on the position of carbon and hydroxyl group. Okay, if these positions are changed, the carbon, for example, these positions are changed, it will convert it to ribulose monophosphate. If again these positions are changed, it is again converting into xylulose 5-phosphate. Getting? So if the carboxyl group, carbon and hydroxyl group is changes, so what happens? The compound also will be changes. So this is an epimerase process. This is an epimerase process. So 4 molecules of xylulose 5-phosphate is epimerized into ribulose monophosphate. So 4 molecules are converting into ribulose monophosphate, and already from ribulose 5-phosphate 2 molecules are produced here, and this is also converting into ribulose monophosphate. So total how many? Here 4 plus here 2, that is 6 ribulose monophosphate is produced here, and the 6 ribulose monophosphate have to be converted into our ribulose 1,5 bisphosphate. So here it is a monophosphate, so we have to add ATP here. So how many molecules are there? 6 molecules of ribulose monophosphate is there. So we are adding 6 ATP to this process. So it turns into ribulose 1,5 biphosphate or ribulose 1,5 diphosphate. Okay, this process is catalyzed by the enzyme kinase. Totally we are getting again 6 RUPP. So from here the whole process, now we are calling it as regeneration phase. So here what we can see that there are 3 processes happening here. There are 3 processes happening in Calvin cycle. One is carboxylation process, and the next one is reduction process, and the third one is regeneration process, carboxylation process, reduction process, and regeneration process. And in this process how many ATPs entering into the cycle? How many ATPs entering into the cycle? Here 12 ATPs and here 6 ATPs. Total 18 ATPs are entering into the cycle. Total 18 ATPs are entering into the cycle. Whereas here we can see that NADPH. How many NADPH are entering? That is 12 NADPH are entering into in this process. Okay, I'm writing here for your understanding purpose. How many? 18 ATP and 12 NADPH are entering into the cycle. This is what about Calvin cycle. Next class we will discuss about further topics. Thank you.